All right, next up is Julia Grenn, one of my graduate students. And I just want to point out, Julia's put it called water parameters, and we had a discussion about water quality. I just want to point out this is one of those research questions that actually a farmer came to us and asked. And you heard in the panel before, Dewey Hauk from Cypress Point Oyster Company. This is a question they basically said, we want to know what's happening in the bags to the water, what's happening to the water parameters. And so this is work that was prompted from a grower question.
Okay, awesome. Hi everyone, thanks for the introduction, Bill. My name is Julia Grenn, and I am a graduate student at the Virginia Institute of Marine Science, and I work with Bill Walton. And today I'll be presenting on how different husbandry decisions can affect the water parameters inside of your floating oyster bags and baskets. So in recent years, there's been piqued interest in off-bottom production due to the benefits it can provide producers, such as increased product quality, better meat yield, product consistency, among other things. However, in the early 2010s, farmers in the Chesapeake Bay region began reporting mortalities of 30% in one-year-old oysters approaching market size. And this number skyrocketed to 85% in 2014. More than 100 industry cases were submitted to the VIM Shellfish Pathology Lab from Maine all the way down to the Gulf of Mexico. Notably, parasitic diseases such as MSX and dermo or harmful algal blooms did not cause these mortalities. So what could be happening? In many cases, at many of these farms, there were apparently suitable ambient water conditions. However, qualitative observations of these farms suggest that a number of factors vary among and even within farms in the terms of conditions within grow-out bags. The nature of mesh bags and baskets produces these microenvironments that are susceptible to decreased water exchange rates. So for example, parameters inside of a clean, empty oyster bag vary from ambient conditions due to the spatiotemporal lag and this reduced water exchange rate. And these variations could be magnified in a stocked bag by different decisions such as stocking density, biofouling control, and even ploidy. So just an example of this is the picture on the right. It's a mesh bag inside of a cage, and you should be able to see the oysters through this bag. However, it's just a wall of biofouling, and that's just an example of how husbandry decisions can vary based on where you're at. So these mortalities occurring sporadically in space and time could be correlated to the microhabitat of these grow-out bags, which in large part can be determined by the decisions farmers make every day. So keeping all of this in mind, we wanted to see if farmers could impact the microclimate of their bags and if this affected oyster performance. So we formulated 3 questions, the first of which, do biofouling control and stocking density decisions affect water parameters inside grow-out bags? 2, do any of these observed differences in the water parameters correlate to oyster performance, including health and disease prevalence? And 3, what combination of farm practices can growers employ to maximize oyster performance while minimizing costs? So 1, are there differences among treatment groups? 2, how does this affect oyster performance? And 3, what does it actually mean for you on your farm and the decisions and trade-offs that you're making every day? So I partnered with a local oyster farm called Big Island Aquaculture, and they have experienced one of these sudden mortality events in recent years. So I used triploid oyster seed that was about 22 millimeters long, and I deployed that into 36 bags that were randomly distributed, into 36 bags that were put on a paired line, and you can see that in my picture. That's a picture of my field site, and that's what I mean by a paired line. So there are 18 bags per side, and I had 6 different treatment groups that were randomly spread throughout the line, and I have a 3x2 study crossing stocking density against biofouling control. So high, normal, and empty densities against air-dried and not air-dried, so 6 different treatment groups. So the original stocking densities, the highly stocked bags were at 600 oysters, and the normally stocked bags were at 400, and this is just based on what the farmer at this farm had been doing. And we've had 2 splits since then, and now the densities that should be at final grow out, we might do a split depending on how they look, but it's at 120 oysters in the highly stocked bags and 80 oysters in the normally stocked bags. So next up are my methods. So I would go out to this farm twice a month from March of last year through October between 9 and 2 p.m., and it would be every other week. And I would collect water from the inside or outside of these bags. I would have to pull up to the line in a kayak, and you can see that here from the video that's playing. I think I stopped it, but so I pull up there as to not disturb the water parameters within these bags, and then I use a peristaltic pump to pump out about 250 milliliters worth of water, and then I use a YSI to assess those parameters in real time. So I'm collecting dissolved oxygen, pH, chlorophyll, and turbidity values. So after the water parameters are collected, I then flip my bags, for the air dry treatments at least, and they're flipped again every 2 weeks. We can switch that to once a week if necessary, and it's for 24 hours. So all of the following data I'm about to show you was collected in a sampling event on September 19th of 2022, which was 2 days before a split. So meaning that the heavily stocked bags reached 70% full and the normally stocked bags reached 50% full. Splitting was just to decrease the density of the oysters within the bags, as I'm sure a lot of you do. And this splitting is also something that the farmer does at his farm. So first up we have dissolved oxygen and stocking density, and we have stocking density listed on the x-axis, dissolved oxygen along the y-axis, and you notice the letters on top of the bars, those indicate statistical significance. So if they share a letter, they are statistically the same, and if they do not share a letter, so A and B, they're different. So regarding dissolved oxygen, empty bags reported the highest dissolved oxygen, and the stocked bags had lower concentrations. And an explanation could be that the oysters are respiring and using this oxygen within the bags. Next up is pH, and we have an interaction effect of stocking density and biofouling control on that parameter. So the graph is structured the same way, and that stocking density is along the x-axis, pH is along the y-axis. The letters on top of the bars indicate statistical significance. However, I've added an extra factor in here with the color of the bar. So air dried is orange and blue is non-air dried. So first thing we notice is that the empty bags have the highest pH values, and then we move to the middle group of the highly stocked bags and the normally stocked air dried group, and then ending with the lowest values recorded in the normally stocked non-air dried bag. So what these results could suggest is that when the oysters are respiring, a byproduct of that is carbon dioxide, so that could be making the immediate water within these bags more acidic. We're still looking at explanations for why the normally stocked non-air dried bag might have the lowest pH though. Next up is turbidity, and it was also an interaction effect of stocking density and biofouling control. And again, the colored bars indicate air dried or not air dried, and the letters on top of the bars indicate statistical significance. So the highest values were reported in the highly stocked non-air dried group, and then we have an intermediate group of the empty bags, and then, because they are statistically the same as the highly stocked non-air dried, and the lowest group, which is the highly stocked air dried and the normally stocked bags. So one important thing that jumped out at us, or one thing to note, was the highly stocked non-air dried group. And some possible explanations for why the turbidity might be higher in these bags is that your oysters are in a highly stocked, non-air dried environment. So because it's highly stocked, they could be stressed out, so they might not be feeding as effectively or as efficiently. We're also in a non-air dried environment, so there could be more biofouling particles floating around that I could have sucked up with the peristaltic pump. Or we noticed that in all of the treatment groups with a highly stocked non-air dried, Olvo was present in all of those bags, and it wasn't present in other treatments. So those could be some reasons why that's higher. Next up, we have chlorophyll, and it's also an interaction effect of stocking density and biofouling control. And it follows a very similar pattern to what we saw with turbidity, in that the empty bags have the highest values, and then we have in the highly stocked non-air dry group, which is statistically higher than some of the other treatments. And then we have the highly stocked air dried and the normally stocked bags. So lower values inside of these bags could suggest that the oysters are eating, so they're filtering the water, which would lead to decreased chlorophyll levels inside these bags. But reasons for turbidity being high are also similar for chlorophyll, since they're linked. So it could be that in the highly stocked non-air dry group, they're not feeding as effectively or as efficiently. It could be the ova in the bag. We could be picking up extra biofouling particles. Or another alternative explanation could be that the phytoplankton communities within these bags might differ from each other in these different treatment groups. And another VIMS graduate student is looking into that. We have the water samples, so hopefully we can provide some more information on that next year. So what does this data tell us? First of all, there are statistical differences among treatment groups based on water parameters. The results suggest that farmers can influence the water parameters within their floating bags through husbandry decisions. And this could correlate to oyster performance. Second, oysters are residing in these bags in conditions that are lower than ambient levels. So generally, dissolved oxygen, pH, chlorophyll, and turbidity, all to varying extents, were lower inside of these bags. So although these conditions within bags are currently not presenting any alarming values, the data demonstrates that everyday decisions can be used as a tool to potentially enhance production on oyster farms. And we hope to provide producers with better data so they can make better decisions, while also identifying factors that could be driving these spring-summer mortality events. I look forward to assessing the rest of my data and to understand if there are trends between water parameters and oyster performance, and putting that in the context of what it means on farm. So before I conclude, I would just like to thank Bruce Boat, who allowed me to deploy this experiment on his farm, and also my lab mates. In my condition, I can't really do field work, so they have been a huge help. And with that, I listed out my contact information, and I will take any questions.
Thank you, Julia. Anybody have any questions for Julia?
Julia, what are the worst possible conditions that you let bags get to that were part of this initial experiment? And you plan to let it go to a degenerate case in the future where you're just trying to see what kind of effect with respect to mortality happens if bags being ripped up?
Yeah, so we simulated what would be happening on a farm. So the worst conceivable conditions, at least in this experiment on this farm, were 70% full for a stocking density and biofouling coverage that was pretty extensive. I wish I put a picture of that up here for the worst that it would get. But there was a second part to that question, but I couldn't hear it.
Any other questions for Julia? All right, thank you, Julia.